In this video, we will take glycerol frozen stocks of strains we will be using in this laboratory and streak them to single colony. We use a sterile loop to do streak number 1 direct from the frozen stock. We use a second sterile loop to do streak number 2, crossing the initial streaks in just 1 or 2 places. Finally, we use a third sterile loop to do a third set of streaks, filling in the rest of the plate. The third set of streak crosses the second set of streaks in only 1 or 2 places. The effect of this is to dilute the cells. In the area of the first streak, the cells are so close together that they form confluent growth. By the end of the third streak, individual cells are more spread out. Where a single cell ends up far away from any others, it grows into a visible colony after overnight growth. Each single colony therefore comes from a single bacterial cell and is therefore genetically pure. I've labelled 4 nutrient agar plates to streak out 4 different strains that we will use as indicator bacteria in our experiments. The first one is Micrococcus luteus, a gram-positive yellow-coloured bacteria. The others are 3 different strains of Escherichia coli, a gram-negative bacteria commonly used in molecular biology laboratories. DS941 is a lab strain of E. coli that is streptomycin resistant. MG1655 is a wild-type E. coli strain that is not resistant to any antibiotics. The final strain is DS941, but contains a plasmid PSB1C3 that makes it chloramphenicol resistant as well as streptomycin resistant. We will start with E. coli DS941. Here we see the frozen glycerol stock for DS941. Loosen the lid and, using a sterile plastic loop, touch it to the top of the glycerol stock. Replace the lid and use the loop to make the first set of streaks on the plate. Rotate the plate and use a new sterile loop to do the second set of streaks, crossing the first set of streaks. Get a new sterile loop, rotate the plate one last time, and do the third set of streaks, crossing over the second set of streaks and being careful not to cross the first set of streaks. The remaining 3 strains are done in much the same way. Here you can see me doing Micrococcus luteus. Remember to use a fresh loop for every streak and not to push too hard. Micrococcus luteus is incubated at 30 degrees centigrade. We will incubate this plate for 30 hours. The E. coli strains need to be incubated at 37 degrees. E. coli colonies will grow in about 16 to 20 hours. Watch the next video to see the results. Remember to try the Learning Science lab simulation to practice streaking cells to single colony.